Now that we know what's happening, I want to take on why. What caused this event to occur in our patient? And it's not at all a simple matter. If it were, we'd have a cure for cancer by now. It's a very, very complex matter. All of these changes that occur in the cells, the proliferation, the invasion, the metastasis, all of them require some pretty serious changes to the cell's phenotype and the cell architecture. Now, we worked out a lot of what has to happen for the cells to become cancerous. And in 2000, 20 years ago, basically the modern theory of cancer was sort of born in a paper by Doug Hanahan and Bob Weinberg. And they published this picture in that paper. They said, okay, here are the things, the 6 things that we are postulating must occur for one perfect cell, healthy cell, to go renegade and become a cancer cell. And one of them is they have to be sustaining their proliferative signaling. They have to evade growth suppressors, whatever those mean. They have to activate the invasion and metastatic processes. That requires changes in the cells of the cytoskeleton. It requires changes in the way the cell is able to adhere to other cells next to it. There's also this ability. They have to become immortal. Most cells are not immortal. Most cells can only divide 35 to 40 times before they start having trouble and going into this, what we call crisis or senescence, and then later crisis. They have to be able to generate their own blood supply. And I showed you that the cells that invade, the tumors that invade, they actually will grow so big that they need their own blood supply. So they have to be able to do that, and that's called angiogenesis. And they have to be able to resist death. They have to be able to sort of be in a really nasty environment, hunker down, and still be able to survive, where a normal cell would be killed. So all of these things have to happen. If you want to study cancer, you should study all of these 6 things. Exactly what has to happen in order for these 6 characteristics to be acquired. We are not going to have time to do that. I've got half of a lecture here, right? So I can't really go through every single one, but I do want to go through a couple of them. Notice sustaining proliferative signaling. Proliferative signaling means a signal that tells a cell to divide. Now we'll get to that here in a moment. You have seen this before, so this connects to something that we've already talked about in the very first unit. Evading growth suppressors. So there are signals coming from the outside portions of the cells, the outside environment, and also internally, that is suppressing the proliferation and growth of tumor cells. And then all this activating invasion, metastasis, and replicative immortality, and so on, I'm going to pretty much skip. This one also is kind of related to these other 2, which I'll talk about here in a moment. But I really want to focus on this one in particular. So in this case, how is it that these characteristics arise? That's what I want to get into, exactly what is happening to get these characteristics to arise. All right, so given that we know that this is what has to happen, we got to go and figure out, well, where is it that the cell goes haywire? What happened to the cell in order to get it to not work? To understand that, we got to go back to Unit 1 and go back to this cell cycle. Remember, the cell cycle was that a cell divides, brand new baby cell enters into this first gap phase, and that gap phase then will automatically default, goes into G0. So we automatically go into G0, and then we hunker down, and the cell can stay like that forever until it dies. Now, normally cells will do that. If they need to divide, they will get a signal. Here we go. There's the proliferative signaling that I'm talking about. They get some signal from the outside, an epidermal growth factor, or some other growth factor, that tells the cell, okay, you need to start to divide, you need to prepare to divide. But in addition, the cell also has to do this. When the cell's told to divide, it gets the outside proliferative signal, but it also has to turn off the machinery that put it into G0 in the first place. So 2 things have to happen at least. 1, you got to release the brake, and 2, you got to put on the accelerator so you go back into G1 and into S. Okay? Now, that's what this G1 checkpoint is all about. The G1 checkpoint normally is turned to the point where it's blocked. So that's why cells go into G0. And that's because they don't have the proliferative signal, and they do have these growth suppressing signals. The natural state of a cell is to have growth suppressing signals. Okay, so normally then we have to turn those things backwards. So we get into this G1 checkpoint. It checks everything we talked about before, goes into the S phase, doubles the DNA, goes into the second gap phase, gets prepared to divide, enters into mitosis, and we also have these other checkpoints. But this is the key one that I want to focus on now. This is the one that we can get a sense for how it is that cancer arises by studying how is it that cancer gets out of G0, because it has to. And what they're saying here, what Hanahan and Weinberg are saying here is this. Two of the things that have to happen in order for a cell to become cancerous is, first of all, this proliferative signaling has to be sustained even when it's not supposed to be there. Again, normally a cell doesn't get a signal to divide, and so therefore it just goes straight into G0. So it has to get a signal. The cancer cell has to get a signal to get out of G0, back into G1, and then get through this G1 checkpoint. That's the first thing that has to happen, but that's not sufficient, because normally a cell will have this growth suppressor activated so that if something goes haywire and it gets a signal to proliferate that isn't supposed to be there, that's outside of what the normal signal should be, the cell will still stay in G0 because of these suppressors that are naturally constantly active. So, in order for us to get cancer, not only do we have to get a sustained proliferative signal when we're not supposed to, but we're going to have to break this. We're going to have to bust it, destroy these breaks that keep the cell from going into G1. And if we do that, then we'll go back into G1, and then we'll go through this cycle, come back down here, and after we've divided, we will still get a growth signal because we have sustained growth signaling, and we will have no brake. The brake on the car is broken, so we go whipping right past G0 if we're cancer, and we go right back into S. And then we just keep going around and around and around. And that's why the cells proliferate. That's why cancer is always understood to be a proliferative disease, a disease where the tumor cells just constantly divide. Okay, so let's talk a little bit about this. The evading growth suppressors. There is one particular growth suppressor which is absolutely critical, and it was discovered because of a disease of the retina, the eye, a cancer of the retina that often occurred in children, not always, and can sometimes occur in adults, but it appeared to be inherited, and in fact it is inherited, and can be. There's also a large number of cases that are not inherited. But this retinoblastoma is associated with a particular gene, and it was so closely associated with that gene that we call the gene the retinoblastoma gene now. So the key thing, one of the key things, the thing that's always active that you must break somehow, is the system that sends the cell into G0 that's driven by this protein, retinoblastoma protein, which we simplify by simply calling pRB. Okay, so pRB is a protein that somehow is involved in getting cells out of G1 and into G0. That protein has to be broken, typically, or the system associated with that protein has to be broken in order for us to get a cancer. Again, it's not sufficient because we have to get all these other things too in order to get full-blown cancer. But this is what we're talking about here. Okay, so if we look at this, this is a chart that was also published in Hanahan and Weinberg, and it shows at the time, in the year 2000, the known interactions of proteins associated with cell signaling. Now, when we talked about the signaling process, we're talking about proliferative signaling and signaling growth suppressors. So each one of these words here, disheveled and glycogen synthase kinase 3 beta and so forth, all of these words here are individual proteins. And they all interact in various ways. For example, you can get these anti-growth factors that are binding to a particular receptor on the cell membrane, which activates these things called SMADs, which activate this molecule called P15, which will inhibit this combination of cyclin D, CDK4, cyclin-dependent kinase 4. We'll talk about that later, which also inhibits RB. Oh, there's our RB. See that? Now you can forget about all this. You'll learn it later. But right now what I'm getting at is there's a signaling pathway here of interacting proteins that lead to RB. So RB, again, is the thing that we need to turn off in order to get the cell out of G0. And normally that happens through this kind of thing. Now, on the other hand, we have growth factors. So notice this is growth and this is anti-growth. The anti-growth factors come along here and they turn RB off. In other words, the anti-growth factors turn this off. But the growth factors, which come through here, receptor tyrosine kinases, which can do all kinds of different things. Here's one, goes to GRB2 sauce, and it activates RAS, activates RAF, activates MEK, activates mitogen activated protein kinase, which goes to the nucleus and does all kinds of stuff. Okay, again, the details of that we don't need to memorize yet. You will in your future studies, but right now not. What you need to understand though is here is just one of the major pathways that is a growth signaling, a proliferative signal. And here is another pathway that is anti-growth, the suppressor. Under normal circumstances, if this cell is told to get out of G0 naturally, not pathologically, and start to divide, the cell must receive both this signal and this signal at the same time. If it gets only one, then it doesn't get this signal. The pRB will be here to stop this whole system down, just shut it down. So pRB is central. This is a really, really important protein. Okay, now, if you look at this picture, you'll notice some of these words, like MAP kinase, are black. Other ones are red, like RB. Notice RB is red. When they put this together, they made red those proteins which are known to be associated with cancer. In other words, something goes haywire with those proteins that can lead to one of these steps and acquisition of one of those 6 traits that they talked about in one of the previous slides. The black ones were at that time not understood to be associated with cancer. Okay? Now things have changed in the last 20 years. But the point is, notice RB is red. RB is known to be associated with cancer. All right, so that's what I want to look at. RB, if I change this somehow, I can change it so that this signal is no longer functional. And that would be evading the growth suppressor. The growth suppressor signal no longer works. We evade it by doing something bad to RB. So let's take a look at pRB. Remember, pRB is a protein, therefore it's a polymer of amino acids, and here is the structure of normal healthy pRB. And if you look here, we look at the translation. We already know what translation is. This is after the ribosome has read the messenger RNA, it's produced this sequence of amino acids, methionine, proline, proline, and so on. Okay, and so you can see it's got this structure right here that is the normal primary structure. It then folds into the blob. We call it tertiary structure. It performs its function and so on. We're going to talk about what its function is here in just a moment, but what I want to do is make the connection to what we've just been dealing with this entire unit, molecular genetics, because this is ultimately a genetic problem. That's what's happening with cancer. It's a genetic disorder. I'm going to take you back to one of the earliest studies that was done on pRB. And it's one of the studies that actually showed that pRB was associated with retinoblasoma and what the particular changes were that occurred to cause it. And in one particular patient, it was a child, a very young child, that had retinoblasoma. They sampled tissue from the tumor and from the non-tumorous tissue. And the protein from the non-tumorous tissue from this one child was this, exactly the normal, healthy, perfectly normal pRB, the same as what's inside of you. You've got this exact same protein inside your cells. This is the protein that shuts that growth program down and puts us into G0 when it's functioning properly. Okay, so they also sampled the tissue from the tumor, and they looked at the sequence of amino acids on those proteins inside the tumor, and what they found was it was different. And here is how it was different. These were the differences. If you look, all the white amino acids were exactly the same in the tumor and in the non-tumorous tissue. But in the tumorous tissue, these red ones were all different. Okay, so if let's go back and let's just take a look here. Look carefully at that red, and then we go back and see how it's different. Okay, so all of these amino acids had changed. They changed their meaning. So we've already seen this before. This is a missense mutation. The fact that there's a whole bunch of a sequence of missense mutations indicates to us that this is probably a frame shift. Okay, that would be something that we would expect. Now, not only were these all different, the red ones, but if you look at the dark ones starting here, if you look there, notice these are all dark darkened out, and the reason that they're darkened out is because they are missing from this retinoblastoma protein in the cancer cells. So in this child's cancer cells, retinoblastoma protein was different where it's red and missing where it's dark. So we also, not in addition to the missense mutation, we also have a nonsense mutation right here. Somewhere along the line, this protein got truncated, almost certainly because of a frame shift that caused missense and nonsense mutations. Okay, what did that do? Okay, we'll get to that. What I want to get to first is, how did that happen? Why is it that in this individual's cancer cells, they had this difference and all of this missing? Okay, well, where is it that proteins come from? This whole unit has been about that. If you don't remember anything else from this unit, remember this statement. Proteins inside cells come from genes. The genes are there in the DNA that tells a cell which proteins to make and how much of each protein to make. That's what the DNA does. So this comes from a DNA sequence somewhere. We've seen this again. This sequence of amino acids comes from a sequence of nucleotides on DNA. What is that sequence? Well, first we have to know where the gene is. Where is the gene that produces this protein? The answer is we know. We know exactly where it is. The gene that produces the pRB protein is a gene called RB1, and it is on 13q14.2. Now, you know what that means. 13 means 13th chromosome. Q is telling us it's the long arm of that number 13 chromosome. Now, here's the number 13 chromosome. So it's got a centromere way off to one end, and so it's on the long arm of that, and it's in position 14.2, right? So remember that region, band, subband. So that is telling us we know where it is. Can we sequence it? Okay, now we've studied that in lab as well, sequencing the DNA. And that's exactly what the study that did this, that found this out, did. It sequenced the DNA to see if it could find out what it was that caused the problem. Now, here's the sequence that they came up with. This sequence of DNA, starting here, is the sequence that they got from this particular patient, but from the healthy cells, from the cells that were healthy. And you'll notice TT, CC, GG, TT, those are all nucleotides. Notice, remember, this is the 5' end. The way we write these things out, this is always the 5' end. The complementary strand, which would be read AA, GG, CC, AAAA, is not seen, and that would be running 3' to 5' because of anti-parallelism. This is how we write the gene out. In this particular way, each one of these blocks is 10 nucleotides. This number here tells you the nucleotide on the far left of that row. So this nucleotide right here is the 181st. You can verify that 10, 20, 30, 40, 50, 60, 60, 120, and so forth. So you can get all that right there. Now, this is only part of the gene, and the way they wrote this out is they have removed the exons. The gene itself is gigantic, and it has many exons. If I remember correctly, it has 27 or 28 exons. And it's got a lot of these huge introns inside of it. So those introns have been cut out in this sequence. But here's the first page of just the coding region, and the start codon is somewhere in here. I can't remember exactly where we could find it if we wanted. But anyway, this is the first page of that gene with the exons removed. Here's the second page. Okay, follow through. Here's the third page. Notice we're at 1921, goes to 2641. The next page, 2701 to 3541, that means this is the 3541st nucleotide right there. And it continues on. There's the next page, and then there's the next page. All right, so this all right here is the gene with the exons removed. If I were to write the entire gene out, it would take page after page after page, because, again, the introns in this particular gene are gigantic. Do you know what introns are? If you don't know what I'm talking about with introns, go back to the earlier lecture, refresh your memory what the difference between exon and intron is. Okay, so you also know the NCBI website. And this is the NCBI here that is showing you the part of the gene that's critical. What we want to do is to find the reason why it is that normal pRB in our patient looks like this, but in our patient, the retinoblastoma cancer cells always have this mistake down here. And it's somewhere in all of this stuff. It's somewhere here. Okay? Can we find it? The answer is yeah, we can. We know where it is. If we go to the NCBI website, it's already been annotated. That's it right there. There's where the mutation is. Now, if I want to make it simple, this is the simplest way I can express this mutation. If we look here, the pRB gene, okay, the RB1 gene in the cancer cells is this. This page has the only difference, in essence, between the normal pRB, all of this stuff here, and the stuff that causes the mistaken pRB. Do you see it? It's very, very subtle. It's not obvious where it is until we look right down here. And you see right there. If we look there, there's a gap. All right, now, here's 2641. Let's go back up to the page where 2641 is, second block of 2641, and see what's supposed to be there. Go back up to 2641. There's 2641, and you see this should read GGGAC. And in fact, here it just reads GGAC. All right, now, what kind of mutation is that? Right? Well, you should know at this point, you should be able to answer that question. That's a point deletion. What are the consequences of that point deletion? Well, we know deletions will tend to give you frame shifts. Point deletions tend to give you frame shifts. So we suspected it was a frame shift based on what we saw with the protein. But what were the effects? Well, we already saw it with the protein. The effects were missense, nonsense. All right, so that was it. That one missing G is what caused all these problems. Okay, now I'm simplifying a bit. Let me make it a little bit more accurate. If we go back up to the NCBI website, this is where the mistake happens. There's all the G's. And you'll notice that the mistake occurs right here in the intron exon border. The end of the exon is actually part of this mutation, and the mutation is missing one of these G's. Okay, that could be really any one of them, but the issue is that the consequence is as if this end of the exon was missing that G, and then the next exon comes on here. Okay, so in the terms of what the consequences are, it's a little more complicated than what I'm showing in this picture, but it's a logically identical mistake in terms of what happens with the resulting protein. So here we have a single deletion that caused all that nonsense with the protein. And did that cause cancer? Okay, what's the answer? Does that cause cancer? Let's explore that idea. All right, so before we can answer the question whether or not this causes cancer, let's just go back and get the big picture. Remember, what we're trying to do is understand why it is that this one patient's tumor arose in his eye as a child, and he has this cancer called retinoblastoma. And what we were looking at was evading growth suppressors, this one characteristic that cancer has to have, and we were looking specifically at one of the particular pathways here in which this retinoblastoma protein was involved, and we found this mistake in the retinoblastoma protein itself right here in his cancer cells, and it was caused by this mutation, this deletion mutation. All right, whatever that did, it caused the protein to no longer be functional. So this change, this nonsense mutation, was the main problem. It caused this protein to not function properly. What does this protein normally do? What's it supposed to do? Well, normally what the protein does is this. Now, there's a lot going on in this picture, and this is very similar to a picture that is in your textbook. It's 1215 of the Freeman text, and it starts on page 268. Now, this one is a little bit more detailed because it's actually written primarily for medical students, but you can just focus here, starting here, right there where the cursor is. Now, there's our RB. That's our pRB. That's the RB protein. And this is another molecule. It's called E2F. It's a protein, and it's a transcription factor. We know what transcription factors are now. Remember, the transcription factors are proteins that bind to the DNA, attract all of the machinery that makes transcription, and then causes transcription to work. Now, normally, though, RB binds to this transcription factor E2F. When it does so, it suppresses E2F, and therefore you get transcriptional repression, which means simply transcription is not going to happen. It's a little bit like the operator and the repressor protein that we saw in E. coli in the lac operon. In this case, though, this is playing a similar role to the repressor protein, but that works differently. It binds specifically to E2F and keeps it from functioning. So Normally then, E2F is a transcription factor that will turn on genes that lead to cell cycle progression, meaning they go into S, it gets out of G0 into G, and goes into S and starts to go through the actual cell cycle. Okay, so that's what this is normally going to do. So here's how pRB works. pRB binds to E2F, stops that transcription factor from transcribing the genes needed to get out of G0 and into G1. So pRB is the brake. That's the main thing that causes the cell to stay in G0. So even if the cell gets signals to start to proliferate, this RB will keep that from happening. All right, now, what normally then happens is if a cell signal comes in correctly and tells it, no, you need to get off of there, so because the cell does normally need to proliferate, something will naturally come along over here and remove it. It's a CDK. It'll remove the pRB. Exactly how is still a little bit under controversy. But once the pRB has been removed, then E2F is functional, and it'll start to actually make the genes that are required for proliferation. What happened to this poor kid is that that mutation that truncated the end portion of that gene, of that protein, caused this to not function at all. It will never bind to E2F. No matter what other signals are coming through, it will never bind to E2F. So therefore, the E2F is always free to function. And so E2F will always be here. It will never be here. That's a mistake. That's the pathology. Not the whole pathology. It's only part of it. Because remember, in addition to that, you also have to have sustained proliferative signals. And that is another thing. That had to be another gene that got broken. So did this cause cancer? The answer is it's a little more complicated than that. It didn't cause cancer specifically. It was an event that had to occur in order for the cancer to arise, but it wasn't sufficient. It wasn't enough to cause it to occur. Other things had to happen. So cancer from one mutation, this one single mutation, is not sufficient to cause cancer. However, it is sufficient to ruin the function of this RB protein, which is giving the cells one of the traits, one of the 6 traits that Hanahan and Weinberg in 2000 suggested needed to happen for all cancers. Now, it turns out that in 2011, Hanahan and Weinberg updated that paper. And it's been argued about since, but now we no longer have 6 characteristic traits. We have all of these characteristic traits. So there's more than 6. And here's the point. More things than just one mistake have to happen to cause the vast majority of cancers. There may be one where one chromosomal translocation is sufficient. But in all other cancers, we basically have this statement still in effect. And this is Hanahan and Weinberg in 2000. Several lines of evidence indicate that tumorigenesis, the origins of new tumors in humans, is a multi-step process, and they reflect genetic alterations. Now, notice it's in humans. This isn't true in all animals necessarily, like chickens. There's some tumors that arise maybe with only one change. Many types of cancers are diagnosed in an age-dependent incidence, meaning the epidemiology, looking at how old people are when they get the cancers. It isn't what we would expect if it was just one hit, one tumor. And in fact, you can use that evidence to determine how many events have to occur, and it's anywhere between 4 and 7. I've seen other types of arguments up to 20 events, genetic events that have to occur on average for the cells to become one renegade cell and become a cancer cell. So what's interesting here is that even though this one mutation is not sufficient to cause cancer, it is a requirement. We have to do something to shut that system down, or at least to shut the evasion of growth suppressors. You have to cause that evasion of growth suppressors. And notice how complicated this is. Now, you're going to study this in your upper division courses and study it in detail to see how all this stuff works. I'm not going to get into that. But just recognize the complexity. And this is a greatly simplified system as well. So as complex as this is, what that tells us is there's many ways where you can break it. You can break this with all kinds of mutations in all kinds of different things. The cyclins and CDKs and all kinds of other things can be mutated and cause problems. In tumor cells, it's often RB or the CDK that is broken. It doesn't work. And you also get too much, sometimes too much of the cyclin D, and sometimes too much of the CDK4 or 6. And those kinds of things then are events that can have a similar effect. So my point is it's complicated. It's a complicated disease. Not every cancer has a broken pRB. This patient's cancer did. And approximately upwards of maybe a third to a half will have mutations in pRB, but most of them have mutations somewhere in the system which blocks this whole process. So again, there are many events that have to occur in order for cancer to arise. This is only one. In the case of this child, other things had to have occurred in order to get the retinoblastoma to get started. In the case of Mary, coming all the way back to the very first patient that we looked at, it wasn't just one mutation in one of her cells that caused her cancer. It was many mutations. What caused them? It was her smoking. The smoking does two things. It causes the cells to reproduce more rapidly, mitogenesis, and that is a way in which you introduce mutations. And another way is for the chemicals themselves to interact with the DNA and mutate it directly, and those are called mutagens. So this is our current theory, and here's where it's encouraging to know this kind of thing. Notice in this particular picture, each one of these little sectors represents one of the characteristic phenotypes that cancer cells acquire. Notice in these things, in these boxes, these are all drugs or classes of drugs that we can use to attack every single one of these characteristics that cancer cells have. We can indeed cure many cancers. We are becoming extremely good at dealing with certain types of cancer. We're awful at dealing with others. Lung cancer, one of them. We're getting better. But the point is, there's a lot more to be done, which is why we're training you so seriously.